I think that we are ready.
Okay, hello everyone. Welcome to session 2 of the second day for the first virtual meeting of Systematic Biogeography and Evolution. So it is a pleasure to be here today to introduce a series of 6 talks in the Symposium of Phylogenomics and Molecular Evolution. First of all, we would like to thank Dr. Remco Bouckaert, Dr. Tauana Cunha, Dr. Mark Springer, Dr. Robert Lanfear, Professor Craig Moritz, and Dr. Irene Julca to accept our invitation and for your participation in this meeting. First of all, this symposium streams live on YouTube, and also we are using Discord for our poster session. As you will see, there are text chat rooms to interchange ideas about the talks and poster in each of the symposium. I will attach through chat the Discord invitation link. So please, we invite the attendees to join our Discord account and search for our different channels. Please use the question and answer comment box to ask questions. This option, as you can see, is in the bottom of your Zoom webinar screen, or you can use our Discord chat room too. We will read all of that question after each talk in order to be answered. Some notes that we have before to start these talks. First of all, we invite all the participants to have into account the code of conduct to interact with other people in Discord and in Zoom. Please make sure to mute your microphones and turn off videos during other people's presentation. Attendees have their microphones and video off by default. So now I would like to introduce our first speaker, who is Dr. Remco Bouckaert. He is a senior research fellow in the Department of Computer Science at the University of Auckland. Dr. Bouckaert is an expert in Bayesian phylogenetics and phylogeography. He is the main software architect of BEAST2 and co-author of the book Bayesian Evolutionary Analysis with BEAST. His research has been focused in developing new statistical and computational techniques with application in bioinformatics, evolutionary biology, and cultural evolution. Today, he is going to talk about an efficient Bayesian multi-species coalescent model, which is implemented in BEAST2. Thank you, Dr. Bouckaert, and welcome to the SBE meeting.
Thank you very much. Can you enable host?
Okay.
Okay, thank you very much for inviting me and giving this talk. I'm not a lecturer, so this is the first time I give a lecture online. This is not sort of what I've been used to. So what I would like to talk about is the following things: the multi-species coalescent. I'll first tell you a little bit about what it is and some of its properties and why you want to use it. And so we have multiple implementations in BEAST. I'm going to first talk a little about the star BEAST family of implementations, and then I'm going to talk about the snap and snapper implementations, and tell you a little bit about how BEAST works with these things, and I finish with some final observations. Okay, so Multi-species coalescent. I've got all kind of stuff in my screen. Sorry. So the multi-species coalescent assumes that you have a species tree. So that's the white tree here. And inside that white tree, you have the gene trees. So the gene trees are restricted to be inside the species trees. At the bottom here, you have the species labels. And then for every species, you can have one or more samples from every of those genes. So in terms of parameter space, so we have one species tree, and we can have one or more gene trees inside that species tree. What happens inside every branch in the species tree is a coalescent process. So the genes follow the process, and that process is driven by the population size. So here the widthness of the branches indicate population sizes. And then for each of the tips of the gene trees, there is sequence data, and that data will be used to infer the gene trees using some site model. So the site model has a number of parameters, like if you use an HKY model, you would have kappas, and other models have more other parameters. And then, so different gene trees can have different relative rates. You can imagine that if you have one locus for a mitochondrial gene and one from a nuclear, they will have quite different speeds in evolution. So you want to have some sense of relative rates between them. And then optionally, you can have relaxed clocks. So it's possible that some part of the tree evolves faster than other parts of the tree, and then you would have branch rates. Now, in Beast is based on Bayesian, doing Bayesian inference. So you would have on top of that set of parameters that you would have priors. And then if you have priors like the U prior that governs the species tree, you would have hyperpriors like the birth rate for the U prior. And so for each and every of those things, you would have to specify what you think they will be. Now, if you look at the multi-species coalescent and compare it with other ones, most methods just concatenate so the sequences. So you would get one big alignment, and it assumes that that alignment follows the same history. And here you can see what happens with the error in shallow gene trees, species trees. So when we say shallow, I'm talking about a few million years, and deep is tens of millions of years. And you can see so that the multi-species coalescent, the red ones here, do much better in terms of error than the other methods. And they keep on improving when you add loci, while many other methods just hit sort of an error bound. That's different with deep phylogenies, where both trends follow more or less the same pattern. And the reason for that is that the gene trees are pretty close to the species tree when you have deep phylogenies. So if you look at the edge lengths, sorry, so that previous one is a measure of how much the species tree differ from the ones that you expect there. And so here we look at the error in the length of the edges that go towards the tips. And again, you see with shallow histories, shallow phylogenies, you get the multi-species coalescent outperforming the concatenation methods. And again, you see this thing where there's a sort of an it converges to an error bound and never gets back even when adding more data. Again, with deep phylogenies, that's slightly different. And here, if you look at the tree topology, with yellow, you see that sort of if you have one locus per species, then it's okay. If you have two individuals per species, you're doing better with the multi-species coalescent. And sort of with the deep one, you see sort of a similar thing. Okay, so when do you want to use the multi-species coalescent? So I'm going to argue always. And you can sort of see in this, so the white species here, tree here on the right, which is the true species tree, that all the gene trees can only start coalescing once they get above the species level. So in here, this red tree, you see that it's always giving slightly older coalescent times than the species tree will. And yeah, there's no other way around it. And then sometimes when you have incomplete lineage sorting, so then that coalescent event can even be much, much older. So the consequence of that is that when you do not use a multi-species coalescent and you assume that the gene tree is equal to the species tree, you get basically the wrong timing. There's some exceptions where I would say you wouldn't want to use the multi-species coalescent. That is when all the loci develop completely dependently. So that is when you talk about viruses like corona, hepatitis B, HIV, and many bacteria are like that. And then, of course, the gene tree and the species tree will be the same in that case. The other cases where you might get away with not using the species coalescent and still get accurate species trees is when you have questions where the dating does not need to be very accurate. So typically the topology can become quite accurate by adding more and more loci even when you concatenate. So if the topology is what you're interested in, then that should be fine. And again, as we just saw, if you have very deep phylogenies, then the coalescent process that happens out here, this time between the split of the species tree and where the gene trees coalesce will be relatively small with respect to the rest of the size of the tree when the tree is very deep, and therefore timing of the gene trees might be close to that of the species tree. All right, so now there's a whole bunch of implementations in Beast that use the multi-species coalescent. So there's a list here. So which one do you want to use? I try to make sort of a little decision graph in which you can sort of determine how to do this. But this is sort of, this decision graph applies, I think, in most cases, but it's not set in stone. So you have to apply your own judgment a bit. But in general, sort of the difference between the star beast family and the snapper family is that the star beast ones have full alignments for every sequence, and with snapper you would have just one SNP or ALP sample per loci. All right, so that's sort of the big split. And then say you have alignments, sequence alignments, then the question is whether you want to have relaxed or random local clock on the species tree. So you can have relaxed clocks on the gene trees, but that would introduce quite a lot of parameters. Having it on the species tree means that it's not going to be taking up that much time, that much extra. Now the thing is, we have a Starbase 2 and just released now Starbase 3 available. But this Starbase 3 is still in development, and Starbase 2 is, has been around for a little while now and is doing very well. Starbase 2 will be slightly slower than Starbase 3. So it depends a bit on whether you need your publication to get out and the analysis done right now, or can hold up a little bit more. So if you're not interested too much in the clock things, are you interested in population sizes? So in Starbase, there are versions of Starbase 2 that integrate out the population sizes, so you don't see them actually, so you cannot log them. Starbase and one other version of Starbase 2 does that, and again Starbase 3 is doing that a bit faster. If you have significant migration events, you might want to look into denim or AIM. And so if you have integration or hybridization, so that means when you have many migration events essentially, all at the same time, then you can have a look at species network. Now, species network is not scaling too well at the moment, is my understanding. So it depends a bit on how many species you have. If you're interested in species delimitation, there is Dissect, and Dissect uses MCMC to jump around the state space with more or less species, so it can merge sets of samples into one species or split them up. The alternative is if you have explicit hypothesis, you can always use any of the other Starbase or even SNAP methods, and then do model comparison, where you do either nested sampling or other ways of doing model comparison. And if you don't want to do anything like that, and you want something that runs fast, use Starbase. Now, if you have SNP data, then sort of there is a nice implementation called SNAP that integrates out all the population gene histories. And unfortunately, we did our best to make it as fast as possible, but it's still quite slow. And then Snapper is a diffusion approximation of SNAP that's actually doing quite well, and it's also just released. The paper was published last week or the week before in SysBio. Okay, now let's have a look at the Starbase family of implementations. So we have a second set of sequence alignments, one for every individual. And so Starbase and Starbase 2 are full Bayesian implementation of a multi-species coalescent. And on top of that, they allow population sizes to change linearly through time. So for example, if you look at this branch here, we start with a population size proportional to the bottoms here, and it grows or it shrinks linearly to something that goes at the top here. So one of the questions you can ask is, if you want to add more data, how is that going to improve the inference? So there's three ways of adding data. You can either add more loci, so add more colors of trees, if you like, in this picture here. You can add more loci, so you can add more sort of samples for the existing genes. So like here, there's only 2 blue genes samples, but you can have 3, 4, or 10. And the other thing you can do is have longer sequences. So here it shows the error that you get, or the shrinkage in error that you get when you add more loci. So both scales here are log scales, and the relation is more or less linear for the speciation times, the times of the splits in the tree, and also for the population sizes, it's best less pronounced. And sort of on the left here, you see the actual error. On the right here, you see the interval sizes, and both of them shrink. So the lower is better. So effectively, what you see is if you double the number of loci, you shrink the error by a linear amount. if you use longer sequences, so the error in the speciation times sort of still decreases, but that in the population size has sort of a lower bound. And the reason it gets a lower bound is that increasing the sequence length means you don't increase the number of coalescent events. But the way to estimate the population sizes are determined through here is through where and how many coalescent events there are in a particular species tree branch. Increasing sequence length does not increase the number of coalescent events, so it will not help up to a certain point. And if you want to add more individuals, so more blue, more green, and more red ones here, you see it shrinks the error, but not as linearly as by adding loci. So in general, what you want to do is if you want to add more data, you want to add more loci. But then we run into the computational issue, and that is that this implementation tends to be quite slow. Now, Starbase 2 is sort of an implementation that adds few things. First of all, it adds relaxed and random local clocks on the species tree instead of on the gene trees. Starbase can only add relaxed clocks on the individual gene trees. And so this helps reduce the size of the state space. Second, it has a computational trick where it integrates out the population sizes. So if you're not interested in population sizes, that really helps in getting the MCMC run much faster. It also has a bunch of new MCMC moves that move both the species and the gene trees at the same time. So you can imagine if you have lots and lots of species, gene trees, and then there's going to be very, very little space for the species tree to move. Because there's always going to be one gene tree that will then fall outside that species tree. So there's a bunch of smarter moves that moves first this proposal for the species tree and then adjusts all the gene trees. And that also helps quite a bit. So in total, we get an order of magnitude faster MCMC with Starbase 2 than with Starbase. And sort of here you can see some results in terms of effective sample size per hour. The red one here is with, these ones here are with Starbase and here with Starbase 2. So you can see that increases quite a bit. This is just for simulated data, but the same holds for some of the real life data sets that Hugh looked at. Okay, now we get into Starbase 3. Because Starbase 2 is, even though it's 10 times faster as Starbase, it's still not fast enough to do full genome inference. And so what we're doing at the moment, we have a project to improve Starbase using a whole bunch of new operators. And so I'm listing here a bunch of things that we have been looking at. I'm not going to go into details too much. It's really just computer science that I'm talking about. One of the issues that we did run into is that because you have so many parameters, you can change one operator, and because there's correlations between the different parameters, you get that if you improve the ESS for one, you can decrease that of another. So there's basically we have to go through a balancing exercise to figure out what the right mixture of these operator is. And yeah, we have not done that yet. But so we started doing that. So here again is ESS per hours, and here there's a whole bunch of different mixtures of operators. And yeah, so as you can see, you get this thing here that massively increases some ESSs, but then decreases others. So yeah, this is what we have to look into. The good news is it looks already that it's going to be an order of magnitude faster than Starbase 2. So that means 2 orders of magnitude faster than Starbase. You just made a release of that. If you want to try it and give feedback on how you go with that, that would be great. So these are the StarBeast implementations. I'm going to talk a little bit about the Snap implementations and also how they fit into these two and how you work with these two. So Snap is a package for doing analysis with SNPs. It assumes that the SNPs that you have in your alignment are independent of each other. So you want to make sure that they're not too close to each other. It only assumes that there's coalescent imitation events, so there's no specialization or gene flow. And so what you end up with is one alignment with all your SNPs. And what Snap is doing is it integrates out all the gene trees. So the StarBeast methods, they don't integrate out the gene trees. You have an explicit representation of the gene tree, and you have to move the gene tree all the time when sampling. Snap does not suffer from that problem. It just integrates them out. So you will never have to look at them again. So there's a full Bayesian implementation. And the biggest problem really is that the complexity is proportional to the number of patterns, the number of species, which is obvious, but then it's also n squared log n in the number of individuals. So that's the number of sequences in your alignment for a particular species. And so that makes that it can handle maybe about 150, or if you are really patient, 200 sequences. But above that basically stops. So now we have Snapper. So Snapper is a diffusion approximation to Snap. And what it does is, instead of having the gene trees here at the bottom of a species tree, it has a distribution over the number of SNPs in that branch. And the distribution of that number of SNPs is informed by your samples. So if you have only one sample, it's going to be very high uncertain distribution with high uncertainty. If you have lots of samples for a particular species, this is going to be very peaked distribution. Right? And then what is happening is that this distribution gets propagated through the tree and gets allows you to calculate the likelihood much faster. So the consequence of that is it can handle much more data than Snap, and so the runtime is not dependent anymore on the number of individuals that you have in your alignment, but it's dependent on the number of species and the number of patterns that you have. And as you can see, the approximation is pretty close. So here we did an analysis. This is analysis with Snap, and this is the same analysis with Snapper. And so what you can see is that if I toggle between the two, there's a slight difference between the two, but it's not really significant, you would say. So good. So how we're going to use Snap and Beast. I'm just going to talk a little bit about the workflow of Beast. So Beast is based on packages. So you would have to install the Snapper package first. There's a package manager that you can access from Beauty. So Beauty is the GUI that allows you to set up analysis and configure everything you want to do with your analysis, set priors and those kind of things. Then you produce an XML file. The XML file goes into Beast. Beast is the workhorse that runs the MCMC algorithm, produces two log files, one with traces in it and one with trees. And then there's post-processing programs like Tracer and Density, and there's a whole lot of other ones that allow you to see whether your analysis is actually valid. Okay, so how are you using Snapper and Beast? So in principle, it's just a matter of selecting the snapper template. Then you input your data. You have to say which sequences go with which species. Then you set up the model parameters, and then set up your priors and MCMC settings. And again, so you do all this in Beauty, produce an XML file, run it in Beast, and then you look in Tracer whether the MCMC converged. The way to do it is you look at the trace. If it looks like a hairy caterpillar, you're fine. If the ESS, if it's over 200, that's a rule of thumb that we always take into account. That's sort of the rough way of testing where your MCMC has converged. And you want to run multiple chains just to make sure that everything goes to the same distribution. And then you can have a look at the tree and see what you're really interested in. So I'm just going to show you a few little things about Beauty. After you've selected the snapper template, you can import your data. So this binary sequence. You can import either binary data, so that will be saying whether the SNP is present or absent in that sequence, or you can use DNA sequences, but then Beast will do the SNP calling for you. And what it does, it looks at the first sequence in the alignment that you give, and then says that's the base that are all the zeros. Yeah, and so there's some facilities to help. If your taxon names are somewhat sensible, you can use some pattern matching to determine what the actual species name is that comes with it. So next is setting up the model parameters. Typically, you would like to estimate the coalescent rate, which is 1 or 2 over the population size. That also means that later on you have to specify a prior on that coalescent site in the priors panel. If you do have non-polymorphic sites, then you would like to include them. So the constant sites are not SNPs per se, but if you, well, it depends a bit on how you collected your data. And it is important in terms of timing. So if you do not include them, then what SNP will try to do is do ascertainment correction. So it will try to estimate the number of constant site based on the well, probability of a constant site, essentially. And so there's a whole lot of ways that ascertainment correction can ascertainment of the data can have an impact on your analysis. So if you know how many non-constant sites there are, then you would like to include them in the analysis. Otherwise, SNP assumes that there is that they are well. I try to estimate them, but it does not take into account things like panels or other ways of getting your data ascertained. So if you get your SNPs based on some standard SNP calling or sequencing how do you call it? Sometimes there are some standards for it, and then you can end up with the constant ones in your alignment. That's just fine. This is an active field of research, and so I'm still work in progress on it. So finally, so next one thing you need to do is set your priors. What we found in general is that the priors that the default priors that are included in the templates are sometimes not very good, especially with SNP. There's a prior that says that lambda has to be some 4 or 5 digit number. That really is, this is one of the disadvantages of doing Bayesian analysis. You have to think about your priors. So one thing you can do is have a very wide prior, but then you still would like to specify this. Like here, we have a uniform prior. So it has this one, the default one, I think of the range of 0, because it's the birth rate, to infinity. And so that's probably an infinite birth rate. Most of the time, you know sort of how well you can actually see from the data how many what the maximum number of mutations is, and therefore how many how high the tree is supposed to be, sort of, from which you can then infer what the birth rate should be. So yeah, if you put a little bit of thought in these, that really helps in getting conversions, but also yeah, it can prevent disastrous analysis. And I mean, one good practice you want to do anyway is to sample from the prior and compare the prior that you get with the posterior. If your posterior is very close to the prior, then it means that the data that you added does not really contribute to anything and that you have not learned very much. If your prior is very wide and get concentrated very much, then you know that that's a contribution from data. So that's that should be more desirable. So finally, you have to set up your MCMC parameters. Now, the biggest one to determine is the chain length, and it really depends on your data how long the chain should be. You don't know that beforehand, so you always have to do some sort of short runs, and based on the short runs, you can then determine how much you need, because you want to aim for an effective sample size of 200 for every parameter that you have, ideally. And yeah, that's something that requires a bit testing out. And it's different for every data set, so it's really hard to tell how much you need. So this 10 million here, which is a default, is something that you need to figure out for yourself how much you need. Right. So I want to make some final observations here. So what are the powers and limitations of multi-species coalescent? So the good thing about multi-species coalescence is it does discover, it's very good at recovering the topology. It's very good in estimating coalescent times accurately, but less so than the topology. And the population sizes comes third in terms of accuracy. Now, there's some difficult cases, so that depends a bit on how your data looks like. If you have lots and lots of samples that coalesce, that are very closely related, you get coalescence very early down in the tree, and it means that there's no coalescent events higher up in the tree anymore. And the consequence of that is that what you're going to get is that the population size for this branch and everything above here they're coming from the prior. So this is something to check when you do analysis, that you see, have a look at what the population sizes higher up in the tree are. And if they are very close to what the prior is, then you know that it might be that some of the branch lengths here are more informed by the prior than the data. So this is the first difficult case. The other difficult case is where all the coalescent events are way out in the root, and then there's no signal, essentially not even signal between species here down in the tree. So you get lots of uncertainty down here. And again, population sizes down here will be determined completely by the prior. So the other thing that can happen is if you do not have enough lineages or not enough loci, then again, so if you have only one gene and only one individual per species, again, you get that your population sizes will be determined by the prior. But yeah, usually it's something in between this case here and in this case. So that's sort of what you would like to aim for. And I suppose this is not too unexpected. It's the coalescent times at the bottom of the tree, so they are more accurate than when you go higher up in the tree. And yeah, so that's obviously when you have data points, the timing here is driven by the sample dates here. Right. So there's some correlations you might want to be aware of. That is that the theta, the population size, and the tree height are correlated. This is just coming from straight coalescent theory. If you have a big population size, like if we take 2 random samples from the participants at the moment, and you look at their most recent common ancestor, that will be coming quite far, quite deep probably, because the people are from different countries. And but if you go in a village out here in New Zealand and take 2 people from the street, there's a higher probability that they just have a much more recent common ancestor. So when you have a larger population size, you would expect larger trees. Now, if you look in your data and you have lots and lots of constant sites, then you would expect smaller trees, right? This is coming from the fact that if you have lots of constant sites, there's going to be not a lot of mutations in the tree. And so the more constant sites you have, the lower the branches, and therefore the lower the height of the tree. Now, if you combine these 2 together, you see that if you have lots of constant sites, you're also going to have a lower theta. So there's an inverse relation between those 2 properties. So one quick comment about detecting anomalies. So lots of things can go wrong in analysis. So what happens when you have sequences end up to the wrong species? So that's something that happened in our initial analysis first. One of the species from C got into B, and as a consequence of that, as you're going to see that the population size blows up. And of course, that is because the wrong assignment means that we're going to have a very deep coalescent event between the wrongly assigned sequence and the other sequences in that set. And yeah, so in general, I would say if you see large population sizes, then have a look at your data, because it might be that you assigned the wrong assignment of the species. Another possibility is that you have cryptic species. So again, you will see large population sizes in that case. And so there's always the possibility of mislabeling. And again, you get large population sizes. Okay, so in summary, I think that we can say that the multi-species coalescent method provides tools for accurate species tree inference. So if the species tree is what you're interested in, then there's almost that. Yeah, I would say, for a few exceptions, the multi-species coalescent is what you want to use. In these 2, there are many implementations. So you want to use the one that is catering for your needs. And again, I would like to point out that Starbeast 2 and Snapper are now available. And so if you want to give that a go and give us feedback on that would be fantastic. So finally, I would like to thank everybody in the Master grant that's working on Starbeast 3 and all previous developers that have been working on these packages. And so there's some links here at the bottom So if you want to see these slides, use this link here. And so all the papers that I've been talking about are linked through there. Okay, thank you very much.
Thank you very much for that wonderful talk. It was very interesting. And again, thank you very much to accept our invitation. Now we will have our asking section. So if any assistant has any question, please, you may take the Q&A feature in Zoom. This icon is in the bottom of your screen. And now Ambrosio will read the question for our panelists, and please, Remco, feel free to check the Q&A screen too.
Okay, we have time for a couple of questions. So the first one is from Monique Romero. She said, very interesting presentation. I would like to ask is there a straightforward way to select loci when you have genomic data, for example, hundreds of thousands loci, to employ a species tree on star beast?
So when you have hundreds of loci, it may be possible with Starbase 3 to actually do all of them. Even with Starbase 2 can go up to, we've done, I think up to 200. It depends a bit on the data, really. So you would like to just give it a try. So, okay, so what the thing we recommend with Snap in particular, because Snap goes slow very quickly if you give too much data, is start with a small data set and just randomly select a number of loci from the set that you have, see whether the data is actually working well with a Starbeast, and if so, then keep on aiming data till you run out of patience or run out of computational resources, essentially. So that's what we're aiming for with the Starbase 3 project, is to allow another order of magnitude bigger data sets than with Starbase 2. But again, so if you have noisy data, typically Starbase tends to be able to have only deal with small data sets. If your data set is very expressive, so your gene trees are very well resolved, then it tends to be possible to do more loci.
Okay, thank you. We have time for we have time for another question. This is from Jimena Grosso. Do you think that future speedups will be because of there are some available smart MCMC moves, or it is more on the architecture of parallel computation?
Well, so what we're hoping for, yes to both. I think smart MCMC moves is one part of the mix. Using parallelization is another part of the mix. But the other part of the mix might be actually looking at different ways of parameterizing the model. So it's possible that at the moment we have one site. So if every locus has a site model with a kappa parameter in it, then if you have 200 loci, that means that you have to estimate 200 kappa parameters. Now, the 200 parameters tend to be, there's only so much values that the kappa parameter can take. So it's possible perhaps to reduce that space of 200 parameters that are completely independent into something that is much smaller. So reducing the parameter space is something that we also want to look into. And so that might actually bring it up another order of magnitude.
Okay, and the last one is from Luz Ochoa. What is the difference between dissect and Stacy? In which cases use each one?
Okay, so from memory, I think Stacy is now retired. first, so the Stacy was not doing. I think it was not doing species delimitation, but I would have to look into that. But I thought it was retired because dissect is doing that much better.
Thank you so much, Dr. Remco.
Thank you.
Cynthia, are you there?
Okay, I'm sorry. So for our second talk, I would like to introduce our second speaker, who is Dr. Tauana Cunha. She is evolutionary biologist interested in the evolution of marine invertebrates. Currently, Dr. Cunha is a postdoctoral fellow at the Smithsonian Tropical Research Institute in Panama. She got a bachelor's degree from the University of Sao Paulo and a PhD from Harvard University. Her research is focused around phylogenesis and the distribution of marine biodiversity in space and time with gastropods, and her group of expertise. Her current work includes resolving Asian divergences, investigating population connectivity, and looking at the genome evolution. So today she's going to talk about congruence and conflict in phylogenomics, inferring Asian gastropod relationships. Thank you, Dr. Cunha, and welcome to the SBE meeting.
Thank you, Cynthia. Can you allow me to share the screen, please?
Yes.
Okay. Thank you, Cynthia, and for the introduction, and thank you, Cynthia and Ambrosio, and the organizers for the invitation and for running a very successful meeting. So today I'm going to talk about congruence and conflict in phylogenomics of gastropods. And this is work that I've done mostly at Harvard with consolidated data. So I want to thank my collaborators and also the sources of funding that made this work possible. And so we all want to understand how our favorite organisms are related and have a nice phylogeny to study them. But both with phylogenetic studies based on the handful of genes, and also even now with genomic data, there are some relationships that we still haven't been able to resolve. And it's common to hear that, that we need more data, right? And this is a sample of papers that I pulled from Google Scholar just by searching for more data and phylogeny, and I included here one of my own. And indeed, more data and genomic data specifically are often needed to resolve some of those hard divergences, and especially for deep nodes. And they have been able to resolve many of the interesting parts of the tree of life and many of those recalcitrant nodes. However, they haven't been able to resolve all of them. And I think this is in large part because of the complexity and the variation in how organisms and their sequences evolve. And so it's important to consider sources of incongruence and of potential error in large data sets, so then we can avoid erroneous inference. So some of the sources that have been widely studied are missing data, and also heterogeneity in substitution rates across sites, across genes, across taxa. And there are several others. So I'm going to mention two more that I'm going to come back to later on in the talk. And these are heterogeneity in the frequencies of amino acids. And this results from site-specific preferences for some amino acids, for one or a small set of amino acids in some sites, but not in others. And we've had for a few years now some very good models that account for the specific type of site heterogeneity. And these models, for example, the CAD model in Phylobase, and we also have some maximum likelihood implementations. These are some of the best models that we have right now. The other potential source of conflict is gene tree conflict, especially from incomplete lineage sorting that I'm going to be talking about more. And we just heard a great talk about the multi-species coalescent and software that accounts for incomplete lineage sorting. And so I focus my talk today on the method side of my work, but I'm actually interested in using those methods to understand the relationships and the evolution of marine invertebrates, and I work on gastropods specifically. So this is a tiny sample of the gastropod diversity in the marine environment alone. There are over 40,000 described species. And if we include also terrestrial and freshwater, it's a really huge group. It's divided in gastropods in general are divided in 5 major lineages. So here we have our turban snails, stop snails, abalones, and keyhole limpets. We have true limpets, we have nerites, we have Can you hear me? I'm sorry, I just got a message that I'm muted.
Yes, we can hear you.
Okay, sorry. So cone snails, moon snails, murex shells. So this group here is where a lot of the shell diversity of the familiar species are found. And then we have our sea slugs, nudibranchs, and most of the terrestrial diversity. And so these groups are not only diverse, but they are also very old. And the divergences between them go back to over 400 million years ago. And so this is probably one of the reasons, ancient divergences plus insufficient data, why this has been historically a very hard problem to resolve. So the strategy here was to assemble a transcriptomic data set with good representation for all of these 5 lineages and apply multiple strategies, multiple methods to resolve those early divergences. So I had different matrices with different gene content accounting for several of those sources of error. And I also used different inference methods. For example, maximum likelihood in IQ3 and Bayesian inference in Phylobase, both using some of the most complex heterogeneous models that account for those amino acid frequencies heterogeneity. And these models are applied in unpartitioned concatenated data sets. I also use maximum likelihood with a partitioned strategy. And finally, summary multi-species coalescent methods. And in these cases, we use gene trees to infer the species tree. And this is because full multi-species coalescent models, like we just said with Remco talk, don't do very well still with very, very large data sets. So here we are talking about many hundreds and a little bit over 1,000 genes. So we use this summary approach. And despite historically being a hard problem, with all of that data and with all of those strategies, I was really happy to find one congruent and well-resolved and well-supported topology across all analyses. So I'm not going to go into the details of the actual tree today, but if you're interested in gastropods, please mention that in the questions or just come talk to me at any time about them. And so this was very nice. We have a good, well-supported framework for studying gastropods, but this is not always the case. It's not always that easy. I focus specifically on this group in red over here. So these are the abalones and the keyhole limpets and top and turban snails. And they pose a very similar problem. So they also have divergences that go back over 200 million years. And the phylogenies that we have so far were largely unresolved at those deep nodes or conflicting between each other. And so I used a very similar approach with transcriptomic data and the same types of methods to try to infer a tree for this group. And what we have here is first this one topology, I collapsed the terminals to the families to simplify the tree. And this is the topology that we recover by using a concatenated analysis with those complex, the best site heterogeneous models that we have available. However, we also recover an alternative topology when using a partitioned strategy and the summary coalescent methods. So just to give you some context here, we have slit shells as the sister group to all other gastроpods, and trochoids as this large diversity here, this large clade. And both of these topologies are very similar for all of the divergences, well resolved, except for two nodes, two specific splits. And this are relative to the position of the abalones and the keyhole limpets. So in one case, in topology 1, abalones diverge early on, and keyhole limpets are part of this clade over here. Whereas in topology 2, abalones are the sister group to trochoids, and keyhole limpets are sister group to both abalones and trochoids. And so the question here was, what is the source of conflict? And the first thing that came to my mind was the method limitation. So each one of those three broad categories has its own limitations. But the first thing that I noticed in particular was that partition analysis and summary coalescent methods both rely on information from individual genes. And so just going over a little bit some of those limitations for each one of those categories. We know that concatenated analysis do not account for gene tree conflict. And in turn, multi-species coalescent methods do account for incompleteness sorting. However, summary methods specifically are prone to error when there is a lot of gene tree estimation error. So here we have increasing amounts of gene estimation error, and we can see how that affects the species tree inference. And finally, regarding partitioned analysis, it has recently been shown that partition analysis, and specifically when we are using site homogeneous models for them, which is the most widely version of partition analysis, which is also the one I use here. So when using those strategies, it has been found that we actually often recover long branch attraction artifacts instead of recovering the true tree. And this is due to an accumulated error over the too many partitions. And so because of that similarity between the summary coalescent methods and partition analysis relying on individual genes, my first hypothesis was that that could be the source of incongruent results, and that uninformative genes and over-partitioning could be behind those conflicts. And so if that's true, we could expect that if we used a set of informative genes only under a full multi-species coalescent model, like the ones Remco just described, we would get topology 1 instead of topology 2. And so to test that, we first need to know what is how well resolved, how informative those genes are. And I did that with likelihood mapping tests. So first we summarized the tree into 4 clusters, and I disregarded everything else that was not relevant to those specific conflicting nodes. And here we have the 3 alternative topologies based on those 4 clusters. So topology 1 and topology 2 are the same as I just showed you from the previous analysis. And so for each one of the genes, we get this likelihood map, this triangle over here, where the coordinates of taxa are distributed position according to how much information they have. So well-resolved coordinates that support either one of those topologies fall into the corners of the triangle, whereas ambiguous coordinates or coordinates that are largely unresolved star-like fall in the edges or in the center of the triangle. So here we are looking at a particular example from one gene that is very little informative, where most of the coordinates are in the center of the triangle. But we also have here an example of a very well-resolved gene where most of the coordinates fall in either one of the corners. And so I did that for about 600 genes. And what I found was that indeed most genes are not informative to resolve those deep nodes, those 2 conflicting deep nodes. So here in the x-axis, we have the amount of resolved coordinates per gene. This is the sum of the corners, so the resolved coordinates here. And we can see that a large number of genes have very little resolved coordinates, and only a few genes are actually very informative. So to move on to the full multi-species coalescent model, I selected the set of 50 and 75 genes that were most informative. And hopefully this also addresses one of the questions that came up for Remco about how to select genes for full multi-species coalescent analysis, and that's a possible strategy. And so to recap, the hypothesis was that uninformative genes and over-partitioning could be behind incongruent results. And so the prediction is that with this smaller set of very informative genes under a full multi-species coalescent model without partitioning, we would obtain topology 1. However, we actually found support for topology 2. And so the same as the summary coalescent method and the partition analysis. So with that, we can conclude that uninformative genes and gene-wise partitioning are not the source of discordance and do not necessarily lead to erroneous inference. But we still want to know what's behind the conflict and hopefully resolve those nodes. And so my next hypothesis relates to biological sources of conflict. Perhaps the most straightforward one to think about is incomplete linear sorting, given that we have two coalescent methods that are giving us one topology, while major concatenated analysis is giving an alternative tree. And this is consistent with the short branches that lead to the divergences that we are discussing, and it's consistent with likely fast divergences for those clades. However, we have another strong candidate, which is heterogeneity, large amounts of heterogeneity in a data set that we know has divergences that are very old and these largest amounts of sequences. So with the concatenated data sets, I used some of those past models, the CAD model in Phylobase and those implementations on IT3 as well. And these models, site heterogeneous models like this, have been shown to actually infer the correct tree even when incomplete linear sorting is present. So even when the generating process includes ILS, site heterogeneous models have been shown to be better at inferring the true species tree compared to site homogeneous models. And unfortunately, at least that I know of, those types of models are not implemented in a framework with the multi-species coalescent. So I ran this StarBeast2 analysis with a site homogeneous model that was available. And so we need implementation of models that account for both sources of discordance and that include both of these perspectives to resolve these and perhaps many other recalcitrant nodes in the tree of life. So what does this mean for the phylogeny of the gastropods? We still have a couple of nodes that are uncertain, and we want to find that out in the future, but we still have a very good framework, which is very well resolved and congruent for all of the analysis for most of the deep backbone of the tree. So this will be very useful for our future studies on the group. And so to summarize, more data is often needed, but not always sufficient, and we should be looking for sources of error, both biological and methodological, if we want to infer the most accurate hypothesis for the relationships of our organisms and also understand the biology behind potential conflicts. I've shown you that most genes are uninformative to resolve a couple of specific conflicts in the gastropod tree, but that these uninformative genes and gene-wise partitioning were not the source of incongruence. And for the future, I really hope to see combined frameworks that incorporate those best models with the multi-species coalescent. And yeah, I think with that, I'll be happy to take some questions in Portuguese as well, if people are there, and in Spanish, and if not here, also later in Discord or just by contacting me. Thank you.
Thank you so much for that amazing talk. And now we will continue with our asking section. So please, if Ambrosio has.
Okay. Yes, there is one question for now. It's from Jimmy Cabra Garcia. Did you analyze your data as nucleotides? If so, how were the results in comparison to amino acids?
Hey, Jimmy, thank you for the question. So no, so all of this work is done on amino acids because those divergences are so deep. So all of this work has been done on amino acids. So we have a little bit more conservation of the sites. So none of those were done on nucleotides.
We have another question from Prashant Sharma. Tauana, which topology is consistent with morphological data sets for the position of Alliotidae, T1 or T2?
Hi, Prashant. Thank you for the question. So actually, I don't remember now if it was a cladistic analysis or based on classification and inference for morphology overall, but topology 2 is the one that is consistent. So Alliotis as sister group to trochoids that has been proposed based on morphology.
Okay, Jimmy made another question, but it's related with the Prashant Sharma question. How different is your hypothesis in relation to a morphological based phylogeny? So I will pass.
Yeah, so I mean, there are more differences. So for this group, there has been quite a bit of work for the gastropods, both morphological and with a handful of genes. And topologies are quite conflicting. So we have a bunch of different hypotheses and proposals. And one thing that I found very common, and perhaps more of the molecular data, is long branch interaction artifacts from the small sampling and sometimes small sampling of taxa. But really, we have a little bit of everything in terms of previous work. But the overall relationships here are also somewhat quite different from what has been proposed before.
There are other questions from Kevin Sanchez. Can you please name the software that implements the evaluation of the informativeness of the loci?
Yes, so I did that in IQ3. So that's a likelihood mapping test, and it's implemented in IQ3. So that's where I ran it.
Okay, thank you. And the last question for Tauana is, there are any gastropod fossil associated in your study?
Hi, Bruno. So not to these two phylogenies. So those were inferred only with genomic data, but I do the work with fossils as well. So I focus on another phylogeny for family level, the keyhole limpets. In there, so gastropods have a great fossil record, is the reason why I chose to work with them. And so for that phylogeny, and that's based on a handful of genes, five nuclear and mitochondrial genes. And in that one, I did incorporate fossils to calibrate the tree. And I've also have work on gastropods specifically using fossils to understand the evolution of the shape of the shell throughout the Mesozoic. And so that's not yet integrated with the phylogeny. So I have work on the morphology of shells and fossils. And I do hope in the future to integrate those two things and to a calibrated tip dating phylogeny for gastropods and to better study their evolution as well. Thank you for the question.
Thank you, Tauana. Now we are going to have a break for 10 minutes.
Thank you. Also, we invite the attendees to join our Discord account and search for our different channels. Thank you for your participation, and we'll come back in 10 minutes. Hi, everybody again. So first of all, thank you again for your participation, and we have some, we would like to report our assistants in Zoom and also in YouTube. Right now we have 218 participants in Zoom and 52 in YouTube. So thank you for your participation. Now for the second section, I would like to introduce our third panelist, who is Dr. Mark Springer. He is an American biologist who has made major contribution to the understanding of mammalian evolutionary history. In particular, he has studied the phylogenetic relationships among and within the other orders in the mammalian tree of life. In addition, he has studied methodological bias in phylogenetic inference, especially those related with incomplete linear sorting, homoplasy, and homology errors, support in species tree methods, and fossil calibration in dated phylogenies. So today, he is going to talk about species tree inference with incomplete linear sorting. And thank you, Dr. Springer, and welcome to the SBE meeting.
Well, thank you very much. And am I able to share my screen?
Yes, one moment. Okay.
All right, are you able to see that?
Yes.
Yes.
Okay. Well, Cynthia and Ambrosio, thank you very much for the invitation. It is a pleasure to be here with everyone. And I'm just going to go ahead and get started. And first thing I wanted to call your attention to is a recent publication that we had in the Journal of Heredity. And much of what I will talk about today is in this publication. So this is also a great chance for me to acknowledge my collaborators, Aaron Malloy, Dan Sloan, Mark Simmons, and John Gatesy. So in systematics, a question we are all interested in: how do we build phylogenetic trees? And this debate in large part has distilled down to a debate about concatenation versus coalescence methods. And if you look at recent papers, you see many papers where both types of methods are employed with phylogenomic data sets. At the same time, I think it's worth mentioning that both of these approaches have potential pitfalls or Achilles heels that can cause them to fail in some instances. This is a concatenation tree that we published back in 2011, showing how different families of mammals are related to each other. So we had 164 mammals, 26 different gene segments. And this has stood up pretty well, but there are also some difficult to resolve nodes on this tree that continue to be thorny problems in phylogenetics. And if we fast forward about 7 or 6 years, 2017, here is another tree for orders and many families of placental mammals that was published by Liu et al. So 82 mammals in this case and some outgroups. And this is a coalescence-based tree, and this is one of the summary coalescence methods, STAR, that was used to infer this tree. And again, this is based on more than 4,000 genes. And in large part, this tree is similar to the tree that we published in 2011, but there are also differences. And the differences tend to occur in regions of the tree where we have short internal branches, some of the difficult polytomies that we are still arguing about today. And we see that across the board with lots of different data sets for higher level relationships among orders and families of mammals. One other feature that I wanted to point out on this tree is toothed whale paraphyly. So the odontocetes, the toothed whales, come out as paraphyletic on this tree. even though there appears to be strong support based on a number of other data sets for the monophyly of toothed whales. So I will come back to this briefly. So with the supermatrix or concatenation approach, we start with individual gene sequences that are aligned, and then we concatenate them all together into one big data set, and we infer a species tree, and we may use different models of sequence evolution for different partitions of the data. One of the major problems with concatenation is that it fails to account for incomplete lineage sorting or deep coalescence. So in the lower right there, for example, you can see that A and B fail to coalesce in their immediate common ancestor, and instead only coalesce much deeper in the tree in the common ancestor of A, B, C, and D. And when we have incomplete lineage sorting and a species tree with consecutive very short branches, as is the case here with X and Y, we can get into something called the anomaly zone, where the most common gene tree, or even more than one gene tree, are more common than gene trees that support the species tree. And with concatenation, the anomaly zone is a potential problem that we need to be very aware of. With coalescence methods, the beauty of coalescence methods is that they explicitly address this problem of incomplete lineage sorting. And there are a variety of different approaches. With the big phylogenomic data sets, the approaches that are used most often are what we've heard a little bit about, the summary coalescence methods. These are most tractable with the large phylogenomic data sets. And with these methods, these summary coalescence methods, such as Astral and Star and MPEST, the basic unit of analysis is what we would refer to as the coalescence gene, or the C gene. So the C gene is a segment of DNA for which there is no intralocus recombination. So we can have free recombination between C genes, but not within a C gene. So the idea here is we take our alignments, we estimate separate gene trees for each of these alignments, and then we infer a species tree based on these gene trees. Well, like concatenation, the summary coalescence methods also have some problems that we need to be aware of, and one of them has to do with the assumption that there is free recombination between C genes, but no intralocus recombination within C genes. And C genes in empirical data sets may in fact be very short, so short that they lack enough phylogenetic signal to actually resolve relationships among taxa that are included in the data set in the gene tree itself. And one of the other points to make here is that C genes get shorter as we add more taxa to a data set because of something that we refer to as the recombination ratchet. So we get more and more recombination breakpoints when we add more taxa to a phylogenetic problem. And in most instances with empirical data, researchers don't address this question with too much interest, it seems, and rather they just sort of assume that the individual gene segments correspond to C genes without actually testing for the occurrence of recombination breakpoints. The difficulty there is that if we have one or more recombination breakpoints within what we think is a C gene, and so that we actually have multiple C genes and we're treating them as a single C gene, then we have a blended history rather than a unique history that is what we're really after with these individual C genes. So that's one issue. A second issue has to do with gene tree heterogeneity. And so the multi-species coalescent, we assume that all gene tree heterogeneity results from incomplete lineage sorting. And as is evident from some of the talks in this meeting, there are many sources of gene tree heterogeneity. And in many cases, these other sources of gene tree heterogeneity may be much more important than incomplete lineage sorting. So that incomplete lineage sorting is sort of dwarfed as a source of gene tree heterogeneity. And I'm not going to go through all of the different types of gene tree heterogeneity that are not the result of incomplete lineage sorting. But I do want to mention a few that you can see in this alignment, which is taken from a phylogenomic data set that was published by Kumar et al. In 2013. So here you can see that there are several taxa that have sequences that look very different from the rest, and that's because these sequences are from a different exon. So we have different exons that are lined up with each other that are not homologous. We also have the tree shrew and the tarsier, where up at the top, and the sequences for the tree shrew and the tarsier for this particular gene are not orthologous with the other sequences and instead are paralogous. And then a third issue that we can see in this illustration is missing data. And so in some cases, this missing data problem is extreme with some of these data sets. And in the most extreme, one of the most extreme examples, the orangutan has only 3 base pairs of sequence out of the entire length of the gene. So this is an issue that we need to be very aware of with the summary coalescence methods. And here I just wanted to get back to the Liu et al. Data set that I mentioned earlier, and just so you can see one more example of this. So if you look at gene 3 out of more than 4,000 from the Liu et al. Data set, you can see that the top 3 sequences, which represent 3 different cetaceans, look very different than the others. And when you look at the gene tree, you can see that the 3 taxa with these sequences cluster together. And so what you have here is one baleen whale, the minke whale, and then you have the baihi and the sperm whale, which are toothed whales, and also the killer whale and the dolphin that are toothed whales. So this is an example of the toothed whale paraphyly that I mentioned. And so what is the cause of this toothed whale paraphyly? Well, you can see the alignment problem up at the top there. And turns out that the sequences for the 3 taxa that are clustered together that might not be expected to be clustered together are sequences for introns. And so we have an alignment in which intron sequences are aligned with exon sequences for part of the alignment. And this results in a long artifactual branch. And there appear to be many genes that are like this in this particular data set. So we think this is what is driving the odontocy paraphyly finding in this particular analysis. So given some of the issues with concatenation and with coalescence-based analysis with sequence-based gene trees, we have become very interested in coalescence-based analyses with retroelement insertions as an alternate approach to phylogenetics or phylogenomics. So retroelements are copy and paste transposable elements, and there are well, one of the reasons that they are useful in phylogenetics is that they are very abundant in eukaryotic genomes. And so they occur typically in the middle repetitive fraction of the genome that you can see in this Cott curve. And so in the case of the human genome, approximately 42% of the human genome is estimated to be comprised of different kinds of retroelements. So there's lots of them. And some of the examples of different types of retroelements include things like sines, the short interspersed nuclear elements like alus, the line elements, the long interspersed nuclear elements, LTR retrotransposons, and also endogenous retroviruses. Getting back to this issue of why retroelements, well, another positive is that we can avoid one of the Achilles heels of concatenation, and that has to do with the anomaly zone. So ILS-aware analyses with retroelement insertions can avoid these anomaly zone problems that negatively impact concatenation. And continuing with this why retroelements question, with retroelement insertions, we can also avoid some of the issues that are problematic for summary coalescence methods with sequence-based gene trees. So if you look at some of the assumptions of the multi-species coalescent, we think that the retroelement insertions satisfy these assumptions much better than sequence-based gene trees. So the first point here is that these retroelement insertions are singular events. So they are presence-absence events, and they're not impacted by interlocus recombination. Second is that they have extremely low homoplasy. So when we find conflicting retroelements, this is almost always the result of incomplete lineage sorting rather than some other cause, such as homoplasy. And then finally, an assumption of the multi-species coalescent is neutral evolution, and we think that retroelement insertions provide a better fit to this assumption than do the typical sort of markers that are used with summary coalescence methods, like protein coding sequences and ultra-conserved elements that might be under strong purifying selection. So given this, we developed a couple of ILS-aware approaches to analyze retroelement data sets. And the first one is what we refer to as astral bipartitions, or astral BP. So on the left here, you can see this retroelement insertion matrix with 5 taxa and 8 different elements. And so each column in the matrix then is zeros and ones, 0 if the retroelement insertion is absent, 1 if it is present. So each column in this matrix can be turned into a gene tree, an incompletely resolved gene tree that has just a single bipartition on it. We can also represent these gene trees with their single bipartitions in Newark format, which you can see down below here. And so if we take a file with the retro elements in Newark format, we can then analyze that with Astral. So we're analyzing the bipartitions with Astral and end up with a species tree and branch links. A second approach is one that we refer to as split decomposition with parsimony, followed by quartet assembly. So this is what we're referring to now as SDP quartets. So the idea here, we can start with the same matrix with 5 taxa and 8 retro elements in this example. We then subsample all quartets of taxa from this matrix. And so with 5 taxa, when we look at all of the possible quartets, there are 5 different quartets. And for each of these quartets, we then use parsimony to find the minimum length quartet topology. These minimum length quartet topologies can be converted into Newark format and assembled with Astral, or we can use a matrix representation with parsimony approach and convert these quartet topologies into an MRP matrix and then analyze that to find the species tree. And so these are the 2 methods that we have used, and I just wanted to finish, or that we have developed and then used with some published retroelement data sets. And I just wanted to finish up by going through a couple of examples of retroelement data sets where we have used these methods. And so the first one is a case of retroelements for paleognath birds, including ratites and tinamous. And so in 2019, there was a paper published by Sackton et al. And a companion paper in Systematic Biology by Cloutier et al., where they described various analyses relating to the phylogeny of these paleognath birds. And so they built a species tree using MPAS. They got the same tree with Astral based on more than 20,000 non-coding loci. And you can see what their tree looks like here. And if you look at the left side of the tree here, near the base of the paleognath radiation, you can see that there are 3 very short branches. And so these are branches that are in coalescent units, and they are very short. And based on these short branches, the authors suggested that this is an example of an empirical phylogeny that is in the anomaly zone. Well, the authors also published a data set of retroelements, including more than 4,000 retroelements. These retroelements were not analyzed in a coalescent framework, and so we decided to apply our new methods and analyze the retroelement insertions. And when we did that, we ended up with a topology that is completely congruent. It's the same topology as for the sequence-based analyses, with the exception of 2 taxa are missing that were not scored for retroelements. So it's the same topology, but where you see a big difference is with respect to these branch lengths. And so if you look at these basal branch lengths, you can see that they are much longer. And in fact, with these longer branch lengths, we are now well outside of the anomaly zone. So we think that other sources of gene tree heterogeneity having nothing to do with incomplete lineage sorting are the reason that we get such short branches on the sequence-based or species tree, where we take sequence-based gene trees and then infer a species tree with MPAST or Astral. And then the final example that I wanted to look at is getting back to mammals. And so in mammals, one of the superorders of mammals is Laurasiatheria. And the Radiatheria includes 6 different orders, 5 of which are shown here. The order that is not shown is the Eulipotyphla. So this is the order that includes shrews and moles and hedgehogs. And the Eulipotyphla is the sister group to the remaining 5 orders. But then relationships among these 5 orders are mostly difficult, and there's a lot of argument about this. Everybody seems to accept that carnivores and pangolins, or Pholidota, group together, but near the base of the tree here, you've got 4 lineages that radiate away from each other very rapidly. So Doronina et al. Published a retroposon data set in 2017, so we decided to analyze this data set with these ILS-Aware methods, and both of the ILS-Aware methods get the same tree. And again, this is a tree based on 139 retroelements. And when you look at the tree, you can see that the branch lengths X and Y are very short. And so with these short branch lengths, we are inside of the anomaly zone in this case. And so with this red triangle indicating the approximate position of where these branch lengths plot for this retroelement based tree. So in one case, we've sort of removed a phylogeny, the paleognath phylogeny, from the anomaly zone. And in another case, for mammals here, we think we have a case where it does reside in the anomaly zone. And so with that, I would like to conclude, and just a couple of conclusions I want to leave you with. First has to do with ground truthing genomic or phylogenomic data, to be aware of problematic sequences, problematic alignments, issues that can cause all sorts of confusion and gene trees that can mess up inferences of species trees. So that would be the sort of the first message. And then the second message just has to do with this debate about concatenation and coalescence analyses based on sequence-based gene trees. We know that there are problems that can crop up with both of these different kinds of analyses. And we view ILS-aware analyses with retroelements as an alternate approach to building species trees, an alternate and potentially powerful approach that can complement these other methods that are used to build species trees. And with the much better genome assemblies that are coming online, we think we will be in a much better position to efficiently extract retroelement insertions from genomes. So with that, I will finish up and I will be happy to take questions.
Thank you so much for the wonderful talk. Now we will have our asking section. So if Ambrosio could read the question.
Okay, we have one question, but I don't know if it's so related with the talk, but it says, is there a known minimum gene length so it can have a decent phylogenetic signal?
So yeah, I think you're getting at the question of how long do C genes need to be in order to get enough signal to resolve relationships. And again, that will depend on how many taxa you have in your data set and whether you're dealing with something that's rapidly evolving or slowly evolving. And you know, I think in a lot of cases we might do a reasonably good job if we've got something like 50 taxa and 1,000 or 1,500 base pairs. But there may be parts of a phylogeny where we have very short branches and there's just no information that's relevant toward resolving those relationships. And so in those cases, as Mark Simmons was discussing, we think that the best approach is really just to collapse those branches. So it's possible to use summary coalescence methods with DNA sequences without actually having a fully resolved gene tree. So if you use Phyml or RAXML, you probably know that when you run gene tree analyses, you get fully resolved bifurcating trees. And we probably don't, well, we don't want that when there's not enough information to actually do that resolution. So it varies a lot. You know, your gene trees, you know, your C genes are going to shrink tremendously as you add more taxa to a phylogenetic problem. So there's not one answer that applies in all cases, but those would just be some of my thoughts on the issue.
Okay, we have another question of Ofelia Delgado. Do gaps in an alignment can be analyzed as it is been done with retroelements with Astral, since you generate 0-1 matrices?
So we think that these ILS-aware methods that I discussed, the SDP quartets and the Astral BP, can also be used with long insertions and deletions. It's worth a try there. Another type of, well, it's actually a type of an insertion, but if you look at numites or numps, whichever you prefer to call them, might be another sort of type of marker that can be analyzed with these methods. So yes, and again, I wouldn't be too keen to use these methods with insertions and deletions that are small. You get a 3 base pair deletion, that may be something that happens very, very commonly, and you get a lot of homoplasy. But if you're dealing with much larger deletions, maybe 75 base pairs, 100 base pairs in length, they may be much, or convergence may be much less common. And so there, I think it would be interesting to apply these kinds of methods. So yeah, very good question.
Okay, we have time for one more question. How conserved are retroelement sequences? Could be possible to design proofs to capture them with an hybridization method?
So how conserved they are, that in large part depends on which family of retroelements you are working on. So if you are dealing with an active family of retroelements, you may have many elements that are all very similar to each other, and they're probably reasonably easy to sort of capture with some hybridization methods. If you're dealing with a family of retroelements that was active 100 million years ago, but is no longer active now, the remaining copies of those individual retroelements, wherever they are scattered in the genome, they've had lots of opportunity to diverge from each other. And it does become more problematic in those cases. Sometimes you can get one retroelement of one type inserted into another retroelement of a different type, and just lots of opportunities for insertions and deletions and so forth. So it is more difficult to work with retroelements at depth. So again, there's no one answer to that question, but those would be some of my thoughts.
Thank you, Dr. Mark. Again, I just want to let you know that some of our attendees are in love with your dog that is behind you.
Oh, yes, good. Well, this is Greta, one of our, we have 2 Huskies and a Malamute. And so whenever I'm in here for a Zoom of any kind, she is almost always in here with me. She's a wonderful dog, Greta.
Thank you so much again for your presentation.
My pleasure.
Really appreciate it.
Okay, thank you. We will continue with our next talk. I would like to introduce Dr. Rolf Lanfear. He is an evolutionary biologist with interest in developmental biology, molecular evolution, somatic mutation, genomics, and phylogenetics. He has an undergraduate degree in ecology from Durham University in the UK, and a master's degree in artificial intelligence from Sussex University in the UK too, and a PhD in developmental biology of cockroaches from the same place. Currently, his research is focused on molecular evolution and phylogenetics. Right now, he's working at the Australian National University in Canberra. Today, he's going to talk about confidence and truth in phylogenomics. So thank you. Thank you, Dr. Lanfear, and welcome to the SBE meeting.
Thanks very much for the introduction, and thanks for the invitation to give a talk. Can everybody hear me?
Yes.
Great. I'll share my screen, and then let's see if this works. Okay, can you see the presentation?
Yes.
Yes.
Great, okay. So thanks everybody. I want to start with an acknowledgement of country. So I want to let you know that I'm talking to you from Ngunnawal country, and I want to acknowledge that the Ngunnawal people are the traditional owners of the land that I'm talking to you from, and I'd like to pay my respects to the Ngunnawal elders past, present, and emerging. And today I want to talk to you about confidence, truth, and I added this and concordance in phylogenomics just to give a bit of a more positive spin. And really this is a talk about some work that we've been doing on concordance factors in my lab at the Australian National University, which is a really lovely place to do research and a great place to do research in phylogenomics. And my colleague Craig Moritz will be telling you things after me. And so if you're ever interested in coming to visit, please do come and visit us. So I want to start with an observation that there's a lot of conflict in phylogenomics. And I'm not talking about conflict between gene trees here, which is what a lot of the previous speakers have been talking about. I'm talking about conflict between studies and sometimes even between people. And so here's an example of what this conflict can often look like. So here's a phylogeny, here's a bird. And if we look at the history of phylogenomics of birds, there's a lot of really awesome studies, and this is one of the reasons I want to use this as an example, is because I love all of these studies. So in 2008, one of the first studies that really called itself a phylogenomic study of birds was published. And it had 19 loci, which these days, that doesn't seem like very many loci, but back in 2008, that really was a lot of data, and 32,000 base pairs of sequence. And this study did a lot of stuff really rigorously. It was really a wonderful study. And one of their conclusions was that because they have more data than everybody else that had come prior to them, and because they used better methods than most people that had come prior to them, they did find some surprising relationships, and these relationships seem to have really very strong statistical support. So they suggested that they were probably right, and that the people who come before them were probably wrong, and that seemed very reasonable. If you wait a couple of years, or 5 years in this case, here's another study with 1,500 loci. These are UCEs this time, which are ultra-conserved elements, which Mark just mentioned. And this is 540,000 base pairs of sequence data. That's a vast amount of sequence data. And again, this is a very rigorous study. It was groundbreaking at the time, and there's a lot of wonderful things. But again, one of their conclusions was we have more data than everybody else, better methods than everybody else. And so they found some surprising relationships with very strong statistical support. And so the claim then is, or the interpretation then is, that they're probably right and everybody else is wrong. The year after that, here's a study with 10,000 loci. So this is an amazing study with almost 50 whole bird genomes published, and I think a whole issue of Science devoted to these bird genomes. And this is one of the papers, which is on the tree of life of birds. Almost half a gigabase of sequence data here. This is just a vast amount of sequence data, just incredible. And again, lots of very rigorous analyses. But again, one of the conclusions of this study was they found some surprising relationships with very strong statistical support. And that probably means that they're right and everybody else is wrong. And then the following year, here's another study, a lot fewer loci this time. These are anchored hybrid enrichment loci, about 250 of them, about 390,000 base pairs of sequence data. The big difference between this study here and the previous studies was they had much better taxon sampling. And so their claim is, well, we've got better taxon sampling. We find a lot of surprising relationships with very strong statistical support. And that means we're probably right and other people are wrong. And so taken on their own, each of these studies is absolutely wonderful, groundbreaking, very interesting and very rigorous. But the problem, and this is a problem that gets repeated across phylogenomics, is if you take these studies as a collection, it's not possible for all of them to be true at the same time. So as a collection, you would see apparently a set of completely mutually exclusive hypotheses. So they agree on lots of parts of the bird phylogeny, but they disagree often on these short branches at the base of the tree. And their conclusions are mutually exclusive. And apparently all of their conclusions have kind of very, very strong statistical support, such that they would all reject each other. And so we get into this kind of situation where we don't know why we end up here. And then we have these, again, very rigorous papers like this one by a lot of the same authors in some of those papers I've already showed you, that tries to ask the question of why do these data sets give us conflicting trees? And this study in particular thought that it was the data type, so whether you're using introns or exons or various other kinds of data to infer your trees. And I'm going to argue in this talk that it's not just the data type, it's other things as well. And before I go any further, I just want to point out that there are many such arguments in phylogenomics. So really you see these arguments with these kind of mutually exclusive hypotheses wherever there's a number of different groups and a lot of interest in the phylogeny of a particular group. So you see it in birds, in mammals, in insects, in plants, and I think almost egregiously sometimes in the deep Metazoan phylogeny with discussions about the relationship between sponges, jellyfish, and sort of the rest of animals. And you see these kind of back and forths, which I don't think necessarily reflect very well on our field, because I think we're failing to acknowledge that we have these sets of mutually exclusive hypotheses that they have to be coming from somewhere. So why is this happening? Well, I think part of it is data type, as the Reddy et al. Paper suggested. But I think another part of it is we often tend to misinterpret this measure of statistical support that we all use called the bootstrap. And the bootstrap is a very useful measure. It's a very important measure, and I'm just going to explain for anyone who doesn't know what it is. I'm sure most people are very, very familiar with the bootstrap, but I'm just going to very quickly explain what it is. So you often see a tree. Here's a tree of 4 cockroaches, and you'll see a number on a branch like 97%, and that's often a bootstrap support. And here's how we calculate a bootstrap. And so I'm going to take my original alignment, which is this one here. So I'll take this alignment, and I'm going to create replicate alignments from this alignment. So I'll sample one site at random from this alignment, and I'll add it to my replicate alignment up the top here. And then I'm going to keep sampling sites at random from my original alignment and adding them to this alignment at the top until this alignment at the top is the same size as the original alignment. So I'll do that now. And you'll see that sometimes I've sampled this site more than once in my replicate alignment, and that's fine. That's what bootstrapping is for. Once I've got my replicate alignment, I then make a tree from that alignment. And now I'm going to repeat this process a lot of times. So I can fit 3 of them on this slide, but typically you do at least 100, often more like 1,000 bootstrap replicates. And then all I'm going to ask is I go back to my original tree and I say, what percentage of my bootstrap replicates contain this branch that separates these 2 cockroaches from these 2 cockroaches over here? And in this case, maybe it was 97%. So that's what bootstrapping is. What does it mean? It's a measure of sampling error of a point estimate. So the point estimate here is the branch. And it's important to remember that it's a measure of sampling error. And one thing that Cecile Anne pointed out to me, I think on Twitter, is that you can think of this a lot like the standard error of a mean. And so the reason that it's useful to think of it like that is I think we're more used to thinking about standard errors of means. And the thing to remember about standard errors is as you add more data, you get less uncertainty. So if you add more data from the same distribution, your standard errors will go down and down. And it's exactly the same with a bootstrap. As you add more data, you get less uncertainty. So with bootstraps, as you add more data, the bootstrap will almost always approach 100%. And what's really important, and I think the thing a lot of us, including me, often forget, is that a bootstrap does not tell you if a branch is true. It just tells you how often it's recovered when you resample the data. And remembering that's really important, because there's lots of ways to get a branch that you can resample lots of times in bootstrap samples, even though that branch is wrong. So let's look at this idea that the bootstrap is the standard error of the mean. So if I had a continuous distribution here and I was calculating the mean, then the mean is obviously a very useful summary statistic of this distribution. But there's something else if you were a statistician. If you could ask for another summary statistic of a distribution, you might be really interested in something about the spread of this distribution. So how far out the tails of this distribution go. And that might be something like the standard deviation. So if I was dealing with continuous variables, which in phylogenetics we're not, at least in phylogenetic trees we're not, then I might like to have something like a standard deviation. And so one question is, wouldn't it be nice to have a measure like the standard deviation of a branch in a phylogeny, some measure of the spread of information that underlies a branch in a phylogeny? And it turns out we've had that measure for a long time, and the concordance factors. So I just want to highlight the work of David Baum, who really, I think, was the first person to call them concordance factors and published this great paper here. And Cecile Anne has done great work in this area, including releasing this software package that you can use to calculate concordance factors. And both of these people have collaborated on various papers, including this one down here. So the work I'm going to present for the rest of this talk is really us building just a little bit on the work that came before, particularly the work of David Baum and Cecile Anne. And the work in this talk is collaboration with Min Bui, who has been a postdoc in my lab for the last few years at ANU and is now a lecturer in computer science at ANU, and Matt Hahn, who was visiting us at the time. And a lot of what I'm going to tell you is published in this paper down here. So I'm going to explain to you how concordance factors work and what they are. And again, I'm sure many people know exactly what they are, but just for those that don't. So in a concordance factor, we take our original alignment, but let's imagine that this alignment was actually an alignment of 3 genes. And so it's not like a bootstrap. I'm not going to resample the data set. Instead, I'm just going to split it up into the 3 genes, and I'm going to calculate one tree for each of the 3 genes. And then I'll just ask how many of my gene trees contain the branch I'm interested in. So in this case, I may still be interested in this one branch down here, but now I want to ask how many of the genes in my collection of genes, imagine I had 100 genes or so, have this branch, and then how many of the gene trees have this branch in them, and it might be 50%. And so the concordance factor has been around for a long time. We implemented it in IQ-Tree to try and get more people to use it, and we in this paper, we solved some problems about taxon sampling. So if you're missing some taxa, how do you account for that? You often, in a large phylogenomic analysis, some taxa will drop out of some gene trees because you weren't able to sequence those genes in those taxa. So we solved that problem, and that's pretty trivial. What we really contribute in this paper, and I'm not going to explain really much about this, is a site concordance factor. So a site concordance factor does the same, does what a gene concordance factor does, but at the level of a site. So what a site concordance factor will tell you is what proportion of sites that could have said something about the existence of this branch in the tree, what proportion of them agree that this is the correct resolution of this branch in the tree. And so we can calculate gene concordance factors and site concordance factors, and we've implemented those in IQ-Tree so that hopefully everyone can calculate these things. And so I want to finish up by showing you an example of why I think it's useful to calculate gene and site concordance factors. So this is a tree of birds, and this is me reanalyzing the data set of Reddy et al., the paper I showed you earlier on. And so at the top, we've got the penguins, and then we've got black-footed albatross, shearwaters, petrels, and fulmars down the bottom. So this is just a section of the bird tree of life that I've extracted from that analysis. And so there are 88 genes in this analysis. And here's a branch that I think you might be interested in. And so here's the branch that groups these 2 penguins. So these are king penguins and emperor penguins up here. And it groups these to the exclusion of everything else. And the bootstrap support for this branch is 100%. And I think if you only calculated bootstraps, you might feel very confident in this branch. You might think, okay, this is a well-resolved tree with a lot of support for this branch. And actually, if you calculate gene concordance factors, it will tell you that almost all of the 88 genes in this analysis also support this branch. And if you calculate site concordance factors, it will tell you that almost all of the sites that could have said anything about that branch, so almost all of the parsimony informative sites about this branch, actually support this being the right resolution of this split here. And so in this case, I think you can be fairly confident that this branch is a real branch in the species tree. There doesn't seem to be, so the bootstrap tells you that when you resample the data, you always get the same answer. And the gene and site concordance factors tell you that this distribution is really very tight, and there's not much spread of information around the support for this branch. But this is not always the way things go. So sometimes things look very, very different. So if we look at this branch down here, so the branch that groups all of the taxa in this group here, including this one at the bottom, all of these taxa to the exclusion of all the other taxa in the bird tree of life, which I'm not showing you here, then again, the bootstrap support is 100%. And remember, I told you that it's very easy to get bootstrap support of 100% because it's asking about resampling. How often do we get this branch when we resample the data? But in this case, the gene concordance factor is just 1%. And this is a little bit more than 1%. It's really just one of the 88 genes in our data set contains this branch. So the other 87 genes in the data set think that these taxa do not group exclusively. These taxa here on the right do not group exclusively to the exclusion of all the other taxa in the tree. The site concordance factor looks a little bit higher, but something that's important to understand about site concordance factors is when there's absolutely no information in an alignment, and when the information is completely random, I mean in an alignment, the site concordance factor will be 33%. So this site concordance factor is 37%, which really means there's a really large spread of information. So that both the gene and site concordance factors are telling you the same thing here, that there's a large spread of information about what the right resolution of this branch is. And so this is like saying the standard error of this branch is very small. So there's very little resampling uncertainty, and that might be expected. 88 genes is quite a lot of data. When you have a lot of data, you get low resampling uncertainty, so high bootstrap support. But there's sort of standard deviation, the concordance factors here are very low, which means there's a large spread of information here. And so I think it's really useful to look at both bootstraps and concordance factors. And I really, the point of this talk is to really encourage more people to do that. So I'll just finish with a few kind of take-home messages here. One is, I think it's important to remember, and I think it's more and more appreciated that this is the case, but I think it's really important to remember that big data sets, so phylogenomic data sets with say 50 genes and up, which is now almost all data sets, you should really always expect every branch in your tree to have 100% bootstrap support. And you shouldn't be surprised when they do. If anything, you should be surprised at branches that don't have 100% bootstrap support, and that should kind of pique your interest. And you should look more carefully at branches with bootstrap support values that are lower than 100%. Gene and site concordance factors really complement bootstraps because they tell you different things. Bootstraps approach 100% as you add more data. It's not universally true because some data sets are very complicated and have kind of very conflicting data that's very finely balanced. But in general, as you add more data, you expect your bootstraps to approach 100%. And that's not true of concordance factors. As you add more data, concordance factors will remain stable. So this is just like the standard deviation of a continuous distribution. If you have enough data, you can estimate the standard deviation pretty well. And as you add more and more and more data, the standard deviation doesn't keep going up or down. The standard deviation will stay about the same. You just get a little bit better at estimating it. And concordance factors are exactly the same. As you add more and more data, your error variance on those estimates goes down, but the estimates don't keep going up or down. So concordance factors remain stable as you add more data. And I really want people to use both. I don't want us to abandon bootstraps. They're very useful. And particularly in phylogenomics, they're very useful if you notice that they're below 100%. But I think adding concordance factors into the mix is really important. They're really simple to calculate in like Q3. We put a lot of effort into trying to make them as simple as possible to calculate. So it's really very little effort to calculate them. And we have a website here that shows you a kind of walkthrough tutorial of exactly how to do it. And finally, I want to encourage people to use networks, e.g. It's like the networks that Claudia Solis-Lemus pointed out to us yesterday with methods like SNAC. Because one thing that concordance factors will start to hopefully tell us and convince us of is that we shouldn't always expect evolution to fit this pattern of just a bifurcating phylogenetic tree. Often it's a lot more complicated and a lot more interesting than that. We know that genes have their own trees, and we often know, as Mark just pointed out to us, that within a gene, there's often lots of trees as well. If there's been recombination in the history of a gene, then that gene doesn't have one gene tree. It may have many trees needed to explain its evolutionary history. And in this case, there's really no escaping the idea that the history of the gene or the clade that you're interested in is really a network and not a tree. So I think it would be useful for all of us to start to think more about evolutionary history in terms of networks than only in terms of bifurcating phylogenetic trees. And so with that, it just reminds me to thank my lab members. This is actually an old picture of my lab from a couple of years ago. And it also remains for me to thank you guys for your attention. Thanks very much.
Thank you so much, Rob, for your talk. And thank you for organize your schedule, because especially with this symposium, we have right now like different time zones. So thank you very much. Now we will have our asking section. So please, Ambrosio, could you read the question for our panel?
Okay, we have two questions and we have time for these two questions. The first one is from Carlos Sarmiento. Would this approach of GCF applicable to partitions besides genes?
Yeah, I think you could use it for partitions. I think that the interpretation may get difficult in some cases, but in general, the GCF still makes, so the gene concordance factor still makes this assumption that whatever you're calling a gene has no recombination in it. It makes the assumption that it's sensible to calculate a single tree for whatever it is you're calling a gene. So partitioned analysis would make great sense, for example, if you had a gene and you knew where the exon boundaries were. And if you called each exon its own partition, then it makes perfect sense to use the GCF for those partitions. But if you're calling codon positions partitions, then the interpretation gets different. Because codon positions are kind of interleaved along a gene, and they're not really kind of the same non-recombining units you might expect. But you may still see GCFs that are lower than you might think, because the kind of biases that affect phylogenetic inference will apply differently to different codon positions as well. So it could still tell you interesting things.
Okay, thank you. And there is another question from Elizabeth Henschel. Great talk. I just would like to know why SCF seems to be frequently bigger, a bigger value than GCF. Also, it is possible to have a low bootstrap value if high SCF and GCF.
Okay, yeah, so the first question was, why is the site concordance factor often bigger than the gene concordance factor? That's because in an individual site, you don't really have much information. So we use a parsimony-based approach, which ultimately results in the lowest, if the information in the alignment is random, the lowest SCF will be about 33%. And it can vary because of sampling error, but it won't go much below 33%, the site concordance factor, because we only really look at 3 possible resolutions after you delete a branch in a tree. Whereas for the gene concordance factor, a gene can give you any tree. We assume there's enough information in a gene, then we can estimate a whole tree. And so a gene tree can give you not just 3 resolutions, but any number of resolutions. And so it's plausible that you can have a branch in a tree that does not exist in any of the gene trees at all. So a gene concordance factor can go right down to 0%, where a site concordance factor really doesn't get much below 33%. And the second part of that question, I already forgot. No, I can't find it on there.
Is that it is possible to have low values of bootstrap values,
yeah.
and high SCF?
So could you have low bootstraps and high? I guess so, but I can't think how. I haven't seen it yet, but I wouldn't rule it out, and I don't know quite what it would mean. I'm not smart enough to think of that on the fly. But let me know if you find it. I'd be very interested.
Thank you so much, Dr. Rob. Unfortunately, we don't have any more time to ask the other to respond to other questions, but maybe if you want to, you can type to respond to other question. Thank you.
Yeah, sure. Thank you.
Good morning. Can you hear me?
Yes, we can hear you, Professor Creel.
Just tell me when you want me to share screen.
Yes. Okay. I would like to introduce our next speaker, who is Professor Craig Moritz. He is an evolutionary biologist who has been immersed in the study of lizards' evolution and speciation. Currently, he is the director of the Center for Biodiversity Analysis and the leader of Evolutionary Biogeography and Conservation Group at the Australian National University. His research is focused on understanding the evolutionary processes that generate species diversity and how this knowledge can inform conservation under rapid environmental change, implementing genomics, phylogenetics, and spatial environmental analysis. Additionally, his lab studies how species respond by migration or adaptation, which is key to finding strategies to promote the persistence of biodiversity. They have been particularly interested in implementing genomics and advanced statistical methods to understand species boundaries and relationships across the radiation of marsupial mammals. Today, he is going to talk about figuring out the tips for macroevolutionary analysis. So welcome, Professor Moritz, and thanks for speaking to us today.
Okay, can I do a share screen now?
Yes.
Excellent. Okay. Morning, good afternoon, good evening, wherever you all are. Like Rob, I would like to acknowledge the traditional custodians of the land I'm on. I'm down at the south coast of New South Wales, which is the Yuin country, and I'll pay my respects to traditional owners, past and future. And I also thank the organisers. I think this is a terrific idea in these COVID times. I really enjoy presentations that I've seen. Can you just confirm that you can hear me okay?
Yes, Professor Gray, we can hear you.
Okay, great. All right, so I want to talk to you today about some ongoing work from a research group. Some of you may know most of my professional career, I've worked on sort of phylogeography and population genetics and thinking about speciation. And just recently, we've decided to think about how better understanding of speciation processes and better sampling of nascent species that might be phylogeographic lineages or whatever can help change our understanding of how speciation drives macroevolutionary patterns. And the work I'm talking about today is very much driven by Sally Potter there on the left, Jason hiding up at the back, Moses Blom, Anna there in black. This crazy photo was taken at a wild party at a herpetology conference, but we don't always dress like this. Okay, so moving on. There's increasingly elaborate and really wonderful models of macroevolution or modeling methods that can be applied to infer something about speciation dynamics over time, how that relates to biogeography, trait evolution. One such example.
I think that Professor Craig
Professor
is having problems with their internet, I think. Let's just wait for 5 minutes, maybe.
Yes, I will try to contact him. If not, it is possible to continue with Irene.
But if you want, maybe it's better to wait some minutes.
Yeah, I think it's better waiting.
Yeah.
Oh, I dropped out. Can you hear me now?
Yes.
Sorry about that.
No, don't worry.
I'll go back to my screen share. This worked flawlessly yesterday. Okay, my apologies. So I'll restart. So almost without exception, these macroevolutionary analyses start with the species as provided by taxonomists. And typically these are morphologically described or discovered species. But we know from a long history of study of speciation that species can arise in a whole variety of ways and typically through an extended or protracted process. So in the cartoon version here on the left, you can see species arising through adaptive speciation, ecologically driven speciation, which can occur very fast even with gene flow, resulting in different phenotypic or different morphologies shown there in the different colored boxes. Conversely, you can have cryptic speciation, sometimes called mutation order speciation, which is simply species establishing genetic incompatibilities, whether through drift or just extended time in isolation and origins of unique mutations. And this can take millions of years. And recently this has been formalized more as a protracted speciation model with different rates of speciation initiation, conversion to full species, and extinction processes, which in the incipient stage, and again, think of a phylogeographic lineage, they can go extinct both through demographic loss, but also through fusion with other lineages, as is well demonstrated in several cases. So this is sort of a modern version of thinking about speciation, or at least how I think about it. And it's driven very much by our empirical observations. So I'm going to talk a lot here about lizards. I have to work on lizards primarily. And in this case, this is a lovely study done by Sonal Singhal, looking at different hybrid zones between morphologically identical, and they really are identical, metapopulations of lizards, where they meet in hybrid zones, and use transcriptomic data to look at the width of clines in the hybrid zones and the extent to which they're coincident in space. That is the center at the bottom right. And what she found was that these indicators of reproductive isolation, how narrow the clines are, how coincident they are, increased almost linearly with divergence time. So in these cases, there is no morphological change. They all have very similar or identical ecologies within species, but their reproductive isolation is scaling in a linear fashion with divergence time, which is no great surprise. That's what you expect from theory under cryptic speciation. So that led us to think about how that relates to species delimitation. And for a long time, people have proposed my apologies again. I don't know why this is happening. It was good yesterday.
No, don't worry.
So in brief, we've used this empirical linear relationship between reproductive isolation and time to propose new species based on this calibration. So this is published a couple of years ago in Systematic Biology, where we elevated some of these highly divergent parapatric lineages to species based primarily on their observed divergence levels or branching times. So we can take that same calibration shown here by this vertical box and apply it to a time tree, in this case obtained from Starbeast, and we use Starbeast to try and get appropriate branch lengths, as explained by Remco earlier today. And if we take these 4 tips at the top, we've subsequently applied high density SNP survey data to these. And you can see, with exception for one hybrid, that these 4 putative taxa, which are deeper, as deep or deeper than the reproductive isolation threshold from our work in the rainforest, are behaving like good species. So we think there's something in this particular system where you've got cryptic divergence, scaling with divergence time, and we can use that to at least propose species boundaries. So we then scaled this up to look at the entire radiation of Euongylus skinks. So there's 122 described species in this group. There are early to mid-Miocene invasion of Australia from Asia. We used custom exon capture and sampled not just the 122 described species, but in most cases, the phylogeographic diversity within them, focusing on fairly deep branches, many of which are confirmed by BPP as just being discrete. So there's about 200 tips in the tree across those 122 species, and typically a couple of individuals for each tip. And then we've analysed that with both concatenation, summary tree methods on the 800 odd loci for which we had complete data. Then after a year of compute time, and Remco, I'm looking forward to Starbase 3, we were able to obtain two representation of Starbase. There's two for independent sets of 100 loci each. And this is by probably an order of magnitude the largest, craziest Starbase estimation ever attempted. So the result was a well-resolved tree. The red dots indicate the particular node among genera where the posterior support is less than 0.95. Everything else is 100, at least among genera. And some interesting biological things came out. We get clade separating across the major Australian biomes, and particularly there's a sort of tropical temperate group here, and then tropical savanna groups here and up here. I won't go into those details. What we were particularly interested in looking at is whether our inferences, or what we could learn more about macroevolution, or the intersection of speciation and macroevolution by sampling down to the scale of phylogeographic lineages within species. So, as I mentioned, we have empirical evidence on the divergence times which beget strong reproductive isolation in this group, and that's shown by this little skinny red line here. And the green tips here are branches that are as deep or deeper than where we see reproductive isolation in hybrid zones. The blue branches are the ones that are shorter. And those also, in some cases, we've got black tips here, which are shorter, and these are ones that are morphologically divergent. There's a whole bunch of them down in here, and have been named as species. We've actually looked for integration amongst those. There is none. So they are good species, but they've got a more recent divergence history than these cryptic lineages, as I mentioned in an earlier slide. So across this 200 odd tips, we have our 120 odd morphospecies, or taxonomic species. We have 38 taxa that are deeper than this threshold, that are strong cases for being cryptic, genuinely cryptic species. And then we've got another 37 or so intraspecific lineages in the sample. And the sampling overall is near 100% complete. So what does this tell us then about macroevolutionary analysis? Well, to start with, we can think of what factors might predispose species to have lineage diversity or strong cryptic diversity within them. And one obvious possibility is just simply range size. Bigger species are more prone to geographic subdivision, and also mean temperature, which has been mentioned previously in the literature. So tropical taxa having more deep phylogeographic diversity than temperate taxa. And both of those are supported in this data set. Turning now to macroevolutionary models, the most obvious thing that would happen when you look at a lineage through time plot. So this is time from the root node to the present. And this is the log scaled number of lineages in those slices, called an LTT plot. You see the black here is sampling just the taxonomic species, and the gray is when you include the tips. Inevitably, when you include the tips, you will get an uptick in the LTT plot toward the present. And this is really where all the information is. As you can see, and as you would expect, they're entirely coincident further deeper in the tree. But at the tips, there's some information here that should inform us about the protracted speciation process, both in terms of time on the x-axis and the difference in net speciation rate on the y-axis. And indeed, when we apply our Panda estimation software from the Moron lab, we do the best fitted models using ARC are different depending on whether you include the lineages or whether you just sample the taxonomic species. So if we sample a taxonomic species, as you expect, given this decrease toward the present in the LTT plot, we infer declining extinction and increasing extinction towards, sorry, declining speciation and increasing extinction toward the present. Bearing in mind that none of these methods can estimate extinction well at all, but at least declining net speciation rate toward the present. Whereas when we include the lineages, the rates are constant. There's a constant turnover of origination of lineages and their extinction through either fusion or demographic loss. And this all pertains to this protracted speciation model. So the Moron group have also recently published this CLAD software, which is very sensitive, or a method for detecting lineage or even tip-specific shifts in diversification rates. So here on the left, I'm showing you the tree, including or sampling all the interspecific lineages or cryptic species. And on the right is the species level tree. And these are colored by diversification rates. So red being faster and blue slower. And here I'll point out that the scales, of course, are different. Here we're going up to about 0.5 on this scale, and here we're getting to about 0.4. So of course, by including all these steps, you're inferring higher diversification rates. That's sort of a mathematical inevitability of this sample. So when we apply
Okay.
Okay, oh,
Oh, again, my apologies. I'm just about done. I really do apologize for this.
no
Don't
worries.
worry.
Beautifully. Say that again now.
Professor Craig, probably you could turn off your video to see if it is.
Yep.
If
Okay, that's a good
good.
idea. Okay, let's try that. So these are parameters estimated by this CLADS model, and there's a whole bunch of them, but what I want to focus on here is just the high-level differences. And consistent with what I showed you with the LTT plots and the Panda results, lineages are more stable toward the present. That is that constant birth-death process. And they also have less variability in rates from parent to daughter. That is more consistent trends in diversification rates than do species. And I should also mention we ran all these analyses across 2 different sets of 100 loci and using both beast concatenation as well as star beast 2. And the inferences actually are remarkably similar, which surprised me. And that's shown here for one set of genes, concatenation at the top and star beast 2 at the bottom. So as I said, we expect the speciation rate, and here we're looking at the tip rates of speciation to be higher in the lineage tree than the species tree, just because of the sampling. But what's interesting actually is the residuals. So which taxa have above average, if you like, increase in tip rates when you include lineages for those compared to those that include species. And this one group in particular sticks out, the Carlia sphinx I'll mention in a minute. And also for cryptoblepharous lizards, they have both the highest tip rates, whether you look at species or lineages. And so now we're starting to think about what's the biological drivers of differences in lineage versus species tip rates. And the residuals are shown at the bottom. Carlia shown as having a higher increase when you include the lineages compared to the background relationship between the two data sets. So I did mention cryptoblepharas here. This is a fascinating group, and the key characteristic of this group is a very recent radiation, and it occupies unique ecological zone. They've moved into the arboreal diurnal lizard niche, which is not really occupied by anything else in Australia, and radiated very rapidly and with sort of mixed processes. These lizards have undergone both cryptic speciation. There's some lineages here within taxonomic species, for example, that are morphologically indistinguishable. And they've also radiated between substrates onto rocks from trees. And you can see this little flat brown rock thing here compared to the grey tree-dwelling skink. And most Blom in my group showed that there was a morphological convergence associated with that, with these parallel shifts across microhabitats. So there's both cryptic and ecologically divergent speciation operating in that group. But the key thing is they've gone very fast because they've occupied a unique ecological zone in Australian squamates. The Kali, which is the other group that's got an exceptionally high rate of lineage formation as well as speciation, is the only group here that has divergent breeding colours between species. And in some experiments, particularly over in this group, it's been shown that does affect female choice. And so this might be, and it is also associated with habitat changes. This group, again, seems to be diversifying very fast, both in terms of cryptic divergence as well as, in this case, changes in signaling colours. So, and again, with apologies for the disruptions due to my internet here, these are some messages. I think it is important if you're trying to associate microevolutionary processes to macroevolutionary outcomes, we need to sample beneath the level of taxonomic species. And that's because speciation isn't instantaneous like macroevolutionary models assume. It's a protracted, extended process, and it's messy. Different tensions of divergence occurring simultaneously, possibly with fusion of populations. So I think this can provide more insights into the link between micro and macro evolution. And in our particular case, it's highlighted several groups of skinks that have interesting biological phenomena that seem to be driving different rates of lineage diversification as opposed to traditional species perspectives. And I'll leave it there with some relief that we survived. Thank you.
Thank you so much for your amazing talk, Professor Moritz. Now we will have our asking section. So I would like to remind you that if someone has any question, you may text in the Q&A feature in your Zoom screen. Now Ambrosio will read the question for our panelists, and Professor Moritz feels free to check the Q&A screen.
Okay, we have one question of Darko Kotoras. Would you say that the species which tend to for highly structured populations tend to occur on highly species clades? I don't know.
I get the question. And hello, Darko. It's nice to hear from you again. Logically, you would think so. I think at the present, we don't have enough evidence to really demonstrate this. So the idea is that species that, for whatever reason, intrinsic dispersal limitation, habitat specialization, or whatever, that forms deeply divided population structure would be more likely to speciate faster. The flip side of that is, of course, highly structured populations are also potentially prone to higher local extinction rates that can reduce the rate of formation of full species. So there are two processes counteracting each other there. So I think we need a lot more work on that question.
Okay, thank you, Professor. We don't have any more questions. So I think that we can pass to another talk. Thank you so much
Okay.
again.
And thank you for organizing this. Cheers.
Okay, let's move on to the last talk. It is a pleasure to introduce Dr. Irene Julca. She is an evolutionary biologist and bioinformatician who finished her PhD at the Autonomous University of Barcelona in the Center of Genomic Regulation in the lab of Toni Galvadon. She is currently working as a postdoctoral researcher in the Mudwil lab at the Nanyang Technological University in Singapore. Her research has been focused on the integration of genomic and transcriptomic data to study the patterns of genes conservation across species with a special focus on plants. Today, she's going to talk about the genomic evidence for recurrent genetic admixture during domestication of Mediterranean olive trees. Welcome, Dr. Julca, and thanks for speaking to us today.
Hi, thank you for the nice introduction. Can you hear me? Sorry.
Yes, yes, we can hear you.
Okay, I will share the screen. Okay, I think it's there. Okay, well, I want to thank the organizers for all the effort to do the meeting. And yeah, I will be talking about a project that we started some years ago. And I will start with an overview. So we wanted to cover different aspects of olive when we started this project. And we started by sequencing the genome because at that time there was no available genome and it was important to have one for our analysis. Then 1 year later appeared another genome of a Silvestris individual, so it was good for our sampling. And this year, just some months ago, it was released another new genome of another cultivar. So up to now, we have 3 genomes and some other resequenced cultivars and wild types. Another aspect that we covered was the comparison of olive with other species. There we cover more or less all whole genome duplications and hybridizations. And we are missing 2 main parts, the comparison of subspecies and cultivars and wild. So for this talk, I will focus in this part, the comparison between cultivars and wild types. As I mentioned, we have until now 46 different cultivars for olive, 10 wild individuals. We resequenced new cultivars, 10 new cultivars, plus the reference genome we sequenced, that is the cultivar Farga, and 1 wild individual. In this map, you can observe the distribution of the different samples. Here, Spain is a bit darker because 5 out of 10 individuals come from Spain. So yeah, we have many samples coming from Spain, but anyway, we have still a good distribution around the Mediterranean basin. One of the main topics in olives is to study the domestication. And since we have a lot of samples, we wanted to understand a bit more this process. We know that there are a lot of studies, a lot of people working on the domestication of olive and using a lot of resources. And the 3 main questions that they try to answer are this one: when the olive was domesticated, where did it happen, and if it happened just once or multiple times. The 2 first questions have somehow accepted answers. So the olive was domesticated around 6,000 years ago. There is 1 accepted place in Levant for a first domestication event, but there is a lot of discussion still for the if happened just once or multiple times. I mean, there is 1 primary domestication event or multiple primary domestication events in different places. In this plot, is just to sum up the main theory. It says that the olive was domesticated around 6,000 years ago in this area called Levant, and then it spread all around the Mediterranean basin. There are other theories that says that it's possible other primary domestication events in the central and western part of the Mediterranean basin. So yeah, before starting, I was talking about wild cultivars and subspecies. So to introduce you a bit more about the species Olea europaea, inside the species we have 6 subspecies. Inside the subspecies europaea, 2 varieties, europaea and silvestris. And the ones that we know that we can see in the markets are the variety europaea, and the silvestris are the wild individuals from which it was domesticated. So yeah, the olives that we know are Olea europaea, subspecies europaea, variety europaea. And I will be talking more about these 2 varieties and less about the subspecies. Okay, so the first thing we done with all the samples we have, since we cover more or less a lot now, was do a SNP calling at nuclear, plastid, and mitochondrial genomes of the genome we sequenced, the cultivar Farga, and we use it as reference. So in this plot, you can see the nuclear SNPs, the heterozygous and homozygous amounts per each individual. You don't have to look at all the names, you just can see the green dots are the cultivars and the blue dots are the silvestris. Here I highlighted the silvestris because it's the silvestris we sequence from Spain. And you can see the amount of SNPs for these individuals is very different from the others. We have a lot of homozygous SNPs and much less heterozygous SNPs in comparison to the other individuals. We have other groups of silvestris here and also other silvestris around here that are more similar in SNP content to cultivars. I highlighted these other silvestris because this silvestris is an important sample coming from Turkey. It's the second olive genome that was sequenced. And as you can see, the amount of SNP is quite similar to the ones, to the cultivars. So yeah, with that in mind, then here we can see the amount of SNPs at plastid and mitochondrial level. It is known that the plastid has 3 main haplotypes, E1, E2, and E3. And they distribute differently in the Mediterranean basin. E1, you find more in the eastern part, where the first domestication happened, while E2 and E3 are more common in the central and western Mediterranean basin. As our reference Farga is from plastid type E3, as we expect, the individuals from this group has less number of SNPs in plastid and mitochondrial. And also there are some individuals with no SNPs, so they have identical plastid type as Farga. And until now, we are quite happy because we have different individuals representing each group. We have wild individuals and cultivars for each of the groups. So until now, everything looks quite fine. Then we reconstructed the phylogenies. We reconstructed first the nuclear phylogeny. And here you can see different patterns. We can observe that the silvestris is divided, the silvestris group is divided into two clusters, let's say. One going out here, closer to the root, to the oat group, and another here, the silvestris, the one from Turkey, that is an important sample for us, is here in the middle of the other cultivars. So for us, it's important, as I mentioned, because it comes from Turkey, and is the individual that is closer to the first domestication event, the wild individual that we have. So yeah, to figure out why this individual is here and what is happening, that took us some time, and we have to explain it, because if not, we couldn't include it in the phylogeny, in the sample. So we have some hypotheses to explain this behavior for this individual. The first one is may represent a very recent domestication. But that is not possible because we know that this is the first domestication event, and all these cultivars here come from that part. So they are very old cultivars, so they don't represent that recent domestication. So we cannot accept that hypothesis. And then we think maybe represent a different primary domestication events. Maybe it was domesticated once here and another time here. But what we will expect if this happens is more a phylogeny like this, where we have sister clades between wild types and cultivars, like wild cultivar, wild cultivar. But in this phylogeny, we don't see that. We see all the cultivars together, more or less, and here one silvestris in the middle. So with that, we cannot accept also that hypothesis. The third one is maybe it's a silvestris that was in contact with cultivars, and for that has some level of introgression. For that, we done more analysis. We also studied the structure of the different samples, and we then accepted two main clusters, genetic clusters. You can observe them in orange and green. And with that, we can divide the samples in three main groups. We have all the wild individuals here. I'm sorry, I don't know if you can see that. All individuals here in orange that are represented only for wild individuals, mainly. And all the individuals, for example, here down, where the other silvestris are represented only by cluster green. And then we have a big group in between composed by a mixture of the two clusters. So yeah, this was pointing out something. So we decided to go for the test of introgression, and we test for integration for all the possible combinations in the phylogeny. And here in this plot, you can see the levels of introgression. To explain, more red means more introgression, less red, less introgression. So we can observe here a different pattern. For example, this cultivar up here looks that has a lot of introgression. And if we do as a map, we can see that the introgression well comes mainly from silvestris from the Western Mediterranean basin. And also, if we observe these other three silvestris that are from different plastic types, we start to think that they are not real silvestris, and they are cultivars that were introgressed by some wild individuals. And we can observe also different levels of introgression in the cultivars in general. But if we look at the group of these silvestris and the other cultivars, we don't see signals for introgression. So yeah, they are really a complete different genetic, they have a really complete different genetic background. And with that, we reject the hypothesis that it's a wild individual that has introgression from other cultivars. And what we think and we support is the hypothesis that this individual was wrongly identified and really represent a feral, meaning a cultivar that went into the wild and become more similar to wild than cultivars. So with that, was very sad for us because we lost the sample that was very close to the first domestication event, and we only trusted the silvestris that are from the Western Mediterranean basin. So we don't have really silvestris samples from the eastern part of the Mediterranean basin. That is a bit sad, but anyway, we can still see some things. Until now, we studied all biparental history of olive. So we also try to understand the maternal side of olive by reconstructing the phylogenies of plastid and mitochondria. And here I'm presenting only the plastid because the mitochondria give similar results. And we included also for plastid some subspecies. So you can observe here in black some subspecies. You don't have to know all the names, just you can distinguish them for colors. And here I sum up in this plot more or less what is happening in this big tree. We have some subspecies dividing the europaea group. We have they cluster by their haplotype, plastid haplotype, which is good. We have the haplotype E3, E2, and E1. But the subspecies appear in between. So we think this is pointing out that Europaea is not a monophyletic group, so has a polyphyletic origin. Also, we can observe an incongruence between the nuclear and the plastid phylogeny. In the nuclear phylogeny, we could observe all the cultivars more or less clustered together, while the wilds were outside as outgroups, more or less. And here we can see that the cultivars cluster in three main groups, here, here, and here, with different wild individuals, pointing out that the maternal side of these individuals come from wild individuals from the Western Mediterranean basin and Central Mediterranean basin, overall in this case. So this is pointing out for a secondary domestication event in these areas. So the hypothesis that we support is that there was a first domestication event here in the Levant, and then there is a secondary domestication in the Central and the Western Mediterranean part of the Mediterranean basin, where the wild individuals were the maternal side and the paternal side come from individuals already domesticated from the first domestication event. And still this process is a process ongoing, so still is happening. They are still, there are still genetic interchange between wild and cultivar, so make it very difficult to study this process. And with that, I want to finish and thank my previous group, especially Tony, Marina, and Pablo, that are following very closely the project. My new group here for their support, and our collaborators, and you for your attention. Okay. Hello.
Thank you so much for your talk. We will continue the Q&A stage. So if someone has any question, you could text in the Q&A feature in Zoom. Dr. Julca, feel free to check the screen. And Ambrosio, I don't know if we have any question from Discord.
For now, we don't have any question, but we could wait 1 minute if maybe someone is typing.
Yeah, don't worry.
Okay. There is a question. Do you attempt any explicit ancestor areas procedure?
Sorry, I didn't hear you well.
Okay. Do you attempt any explicit ancestor areas procedure?
I don't get the question. Like ancestral areas to sample, do you mean? Like we try to sample there? It's quite difficult to sample now in the eastern part of the Mediterranean basin because we had to go to Syria, I think, or this area. So it's not possible now. We don't have samples from there. So, and also I think there are very few wild populations in that areas. And there are more wild populations in the Western and Central Mediterranean basin. So yeah, there are many problems to get samples from there. If that was the question, I'm not so sure.
I'm not so sure too.
Do you think?
I think that maybe the question is like for a technical procedure of maybe reconstruct some ancestral areas.
To reconstruct the phylogenies, maybe.
Yeah, I think that.
Different approaches. We use the SNPs we have, homozygous, heterozygous combinations, and also different positions like for degenerates that in theory they evolve slower, so you could see more difference. And all of them, they gave us the same signal. Also, we use different reference genomes, not only Farga, we use also Silvestris as reference genome for some of the examples. And we always come up with the same topology. So it's quite supported that the results that this Silvestris that we had is not a real Silvestris. And for the samples that we have, all the samples coming from the Eastern Mediterranean basin, they are probably more feral individuals or Silvestris with a lot of contact with cultivars. So yeah, it's not easy to the sampling
Okay.
also.
Thank you so much, Dr. Irene.
Okay, thank you to you. Thank you for all the effort to organize this event.
Okay. We don't have any more questions, Cynthia.
Okay, that's fine.
Oh,
We're actually two people here, so it's not Tauana speaking. So this is Bruno.
go
Oh,
ahead.
yeah. So my question is, so this feral population, do you know any phenotypic differences? Like, how much did they evolve in their long-lived trees? Only a couple thousand years. I was surprised to see there is something that evolves to be feral again. Is it phenotypically different from the wild populations?
Sorry, the phenotypic difference between the populations, the wild individuals.
Yeah. So the secondarily, so the feral population, like, is there anything obvious? Like, did it evolve to be completely wild again, or?
Yeah, no, it's still, as I mentioned, well, the olive trees can live up, according to the people that know them more, can live up to 1,000 years. The olive trees can live hundreds of years. So it's a process very slow, the domestication in these trees. So yeah, there is no much, well, with the cultivars, some cultivars are very well differentiated, but when you go into the wild, it's difficult to recognize a real wild type from a feral, for example. The experts said that, and there are many studies that happen to confuse, that in which they confuse ferals for silvestris. Sorry?
Thank you.
Yeah.
Thank you so much, Dr. Julca.
Okay, thank
Talk.
you.
And thank you for listening and your kind attention. And thanks again, all speakers, for your talks and your time. We really appreciate having had this opportunity to share all that wonderful talks. And also, I would like to invite you to keep on attending 2 more sessions for tomorrow. The first one is the Symposium of Diversity and Phenotypic Evolution. We will have our last talk in the Gender and Diversity in STEM series. And in the afternoon, we will have the Symposium of Paleontology and Phylogenetics. So thank you again for your attention and see you tomorrow.